Hello, my name is Kelly Truax, and today I'm going to be talking about our work using laser-induced fluorescence with vegetation towards an improved imaging technique in pursuit of a portable system. A little bit of background about our project is that we're part of the Thrust Area 2 for the MTV Consortium. We are dedicated to the development of technology and methodology that uses and observes biota. Specifically, we're interested in detecting contamination, nuclear fallout, mining waste, organics, or anything else that might be able to be accumulated in vegetation. We're currently developing a non-invasive remote laser-induced fluorescence technique. Our previous work presented at UPR and at previous MTV annual conferences has focused on using the biofinder, specifically on moss, but we've been working on new experiments to focus on environmental stressors, optimizing different chlorophyll focused laser system, specifically for a field application and potential use with different vegetation. We've been exploring these new laser wavelengths due to their ability to be more cost effective and more useful in a portable system. In terms of mission relevance, the NSA mission focuses on the detection and prevention of nuclear proliferation activities. Our study is developing this remote sensing technique for detecting biological response after exposure of plants to metals of interest. This can be due to mining, nuclear waste, or even a nuclear event. Remote sensing technology is useful because it can be used to identify the presence or type of contamination, to produce maps and spatial distributions of that said contamination, and to pinpoint the source and aid in bioremediation. Our technical approach from this summer looks directly at stressors. We designed an experiment to test multiple conditions for moss exposure to heavy metals, photoperiods, and environmental stressors. The metal results were presented at UPR in Atlanta in 2021, and the experiment was inspired by and improved by Wendy Kune at Savannah River National Laboratory in regards to improving our sensitivity and suggestions regarding environmental impact and variability. We decided to test multiple metals. Previous work had focused specifically upon copper with some light testing of zinc and lead, but we wanted to do it at a higher sensitivity. We also wanted to know the impacts of different photoperiods. Us being in Hawaii, we don't have a large variation in photoperiod throughout the year, but if we were to apply the technique to other locations, it would be good to know if a long photoperiod or a short photoperiod could be impactful. It's also important that we understand the impacts that drought or flood could have on the moss samples, or if there are excess nutrients due to agriculture, what that impact might have upon the imaging system. We did several trials testing the above stressors along with the control, and we're still in the pursuit of the exploration of a best analytical method for image batch processing. The results from this work showed that we had an improved sensitivity to the nanomolar per centimeter level, squared level. Metal concentrations detected successfully. The environmental conditions, which you can see in the middle figure, did not interfere with metal detection and are relatively indistinguishable from the control sample. Photoperiod, on the other hand, especially the long photoperiod or a dark period, an extremely dark period, can have impacts on the color shifting. So further exploration how that might impact what we would see in terms of a contamination is important. There were some challenges in our results in terms of zinc being less reactive in terms of the red color channel. We also had some difficulty with copper, though lead and a mixture of metals were easy to detect. We also ran a second experiment in the fall, specifically to investigate if stress input is strictly chlorophyll related. This is due to feedback from individuals at Lawrence Livermore National Laboratory in terms of understanding if metal interaction was simply with the chlorophyll or if it was due to protein or lipid interactions within the cell structures. So we designed an experiment to test chlorophyll A and B specific lasers based upon the work by Yuta et al. Their work looked at ferns, and you can see in the top right figure that it shows the absorption bands for chlorophyll A and chlorophyll B. We focus on using two different laser systems, one at 445 nanometer, which you can see where the arrow is pointing with the excitation, and a 462 nanometer blue laser for chlorophyll B, which is the red dotted line. The idea being that we would try and do excitation at the largest peak of excitation. We can then do measurement of the emission spectra, which we used filters in order to be highly specific for each individual chlorophyll. For chlorophyll A, that's a 670 nanometer bandpass filter, and for chlorophyll B, that's a 650 nanometer bandpass filter. We imaged whole moss masses, individual mass fronds, and then we finally extracted chlorophyll from the fronds and measured the chlorophyll AB ratios independently using a spectrophotometer. The new system is incredibly promising. Potentially better specificity and sensitivity than the biofinder. It also would be much more power and cost effective if we're moving towards a portable laser system. Here we have some of the results from the testing that we did in the fall. You can see the chlorophyll A laser on the right, the chlorophyll B laser in the middle, and the traditional biofinder that we've been using on the right-hand side. We did the same tests with the copper from this summer, which we had difficulty with getting really low specificity with. We did it with the 1 nanomolar, the 10 nanomolar, and 100 nanomolar per centimeter squared. What we end up seeing is if we use the laser with its specified filter, we actually get a really great response between 10 and 100 nanomolars per centimeter squared. It's a little difficult to tell the difference between the two of them, but the clear separation from 1 nanomolar per centimeter squared, which would be well within background levels of metals, means that we would be able to distinguish between what was naturally in the environment versus a potential introduction by a contamination means. What's interesting is that we see that both of the new blue lasers that are chlorophyll specific do a very good job of picking this out, but the biofinder seems to struggle. We did imaging every 8 hours, and we only did one single dose at the 0 time mark to see how the decay would happen. The difficulty is that the decay happens very quickly here, but we are confident that if there was a repeated dosing at low levels, we would be able to detect that. We also did a chlorophyll extraction, as mentioned previously. We had frond imaging with the laser-induced fluorescence. Then we did extraction so that we could do a comparison directly to each other, with the spectrophotometer being used in order to do specific chlorophyll A, B extraction measurements. What we ended up finding is that if we look at the top right figure with the laser-induced MOST image analysis from the previous slide, compared to the spectrophotometric analysis of the chlorophyll extract, we see a marked decrease in the total chlorophyll content at the exact same time that we do the dosing. This is very encouraging. And we're going to continue doing frond and extraction analysis in order to better understand the chlorophyll pigments and the subtleties that are occurring there. Because of the great work that's happened with this, we've been able to go ahead and deploy both of the laser systems into portable designs. The one on the top right is more of a cell phone design using a cell phone camera with a camera app in order to control the exposure levels or the timing or the ISO of what we're imaging. The one on the top right is a portable tablet computer system that has a rear-facing camera that we could take into the field. The benefit with both is that we could integrate Python and do real-time analysis of the images as they're being collected. We have used the top right system for the images you see below. The plants that you see are a simple lettuce, a spinach, and some microgreens, and we see a really lovely response from those just using the laser system as is. Our expected impact is the possibility of a real-time detection of metal contamination in biota. There are challenges to extensive sampling and laboratory analysis that could be removed by target sampling using this technique to save on analytical costs. The technique is not limited by day or light conditions, as opposed to similar agricultural deployed units like near infrared, and it can be used at the meter scale distance. This new portable unit is quite low energy use, and it's a much lower payload. So the portable design for in situ data analysis is incredibly promising. There's flexibility in the camera type being used for capturing images. We've traditionally used a CMOS camera, which can be kind of expensive and really fancy, but we've been able to show that a cell phone or even a tablet can be really useful with the correct filters. Field testing is ongoing for the handheld units that we're making, with the intent that they could be adapted for remote sensing via drone as the end goal. Thanks to MTV, they've been able to support me and an undergraduate student who had a poster presentation here, and has allowed for collaboration with national laboratories. That's impacted our experimental design through several meetings. We've been able to discuss future collaborations, opportunities for site visits, and potential application or internships in the coming year. We've had the opportunity to connect and network with other groups due to the meeting at UPR in Atlanta. Savannah River has continued to be a source of guidance on contamination levels and possible vegetation applications. Lawrence Livermore National Laboratory offered fantastic guidance and expertise on experimental design, testing of the new laser system, and sample preparation. Collaboration has also occurred on a separate project that's ongoing with Pacific Northwest National Laboratory. In conclusion, the experiments conducted have given us a better understanding of the challenges of application in the field. Environmental stressors and photoperiod length are both considerations that should be taken into account regardless of the vegetation that we're working with the possibility that photoperiod could be more of a challenge than other environmental stressors may be. The new chlorophyll-specific laser wavelengths were able to detect as low as 10 nanomolars per centimeter squared levels of copper. The smaller size and affordable cost of these blue lasers makes them ideal for future remote applications and testing. Further tests need to be done in order to better understand the potential impacts of external light conditions from the sun and to determine the maximum distance and if there should be adjustments made to the laser being used to increase our distance. Laser-induced fluorescence response in moss and their chlorophyll extracts showed promising results and helped to explain underlying physiological changes in moss based upon total chlorophyll and AB ratios. The prototype of the portable system is travel ready, but we really want to do a tremendous amount of field testing in order to better understand our analysis techniques. Our next steps are application to different vegetation types like the azolla and duckweed you see on the right hand side, or like the lettuces or the herbs that we're growing in the laboratory. Finalizing a portable system test in the lab will be important to deployment to the field. Testing for external light input, impact of distance, and then adjusting the laser accordingly. Complete code for field batch processing, and finalizing analysis of frond and chlorophyll extract images to have a more complete picture of the experiments that we ran in the fall. Work with the national labs to optimize our techniques will always be very important and will continue as we go forward. With that, I'd like to say thank you for listening to me talk about my research, and thank you to the consortium and the NNSA and DOE for their continued support. Thank you very much.